Hello students, today I am going to teach you the fourth part of molecular breeding for crop improvement. In the previous classes, we have discussed up to the QTL, that is the quantitative trait loci, and I also discussed about your doubts, and I think there these doubts are clarified. Today I am going to discuss the marker assisted recurrent selection and genomic selection. Basically, the selection of a crop is very much essential to improve its quality and quantity. In case of QTL and marker assisted backcross, we have noticed that we generally select the dominant trait over the recessive trait, and the dominant trait containing plant can be considered as the male, whereas the recessive plant is considering as female. But due to the maternal inheritance property of female plant, it is necessary sometimes that the recessive selection may be required, and thus the marker assisted recurrent selection is very much useful to select those recessive traits. In this figure, what I am describing you, the donor plant is depicted here by red color, and the recessive plant is and the recurrent plant, that is the recessive one or the female one, is represented by the blue color. Upon the F1 hybrid formation, that is the one chromosome comes from the paternal and one comes from the maternal inheritance, and this is the F1 hybrid, which is test crossed with the recurrent parent. The test cross means the cross between homozygous recessive plant. It is this test cross must be a cross between F1 hybrid with the homozygous recessive parent. That means here the female parent. And after that, we can generate the test cross progenies or back cross progenies, and after these progenies development, we use several crossing, that is those are the test crossing, and ultimately we develop a plant that contains the same trait at the same loci of both the chromosomes, and this is our main target. So the markers could be used to assist selection at time where the phenotype selection cannot be performed, and marker assisted recurrent selection is a recurrent selection that utilizes markers to increase gain per unit time. Where the phenotypic selection cannot be performed, then we use the marker assisted recurrent selection. Generally, when we use the recessive traits, the phenotype, the recessive trait is not expressed, and thus it is impossible to select these recessive traits with phenotyping, and thus we use marker assisted recurrent selection. Now, what are the steps in a marker assisted recurrent selection in a crop? First, molecular marker assisted selection in cycle 0. That means we have to select some markers in the 0 cycle. What is 0 cycle? 0 cycle means we first uh, create an F2. That means in the hybrid, that when the two hybrids are crossed, then only the F2 can be achieved. Then this F2 must be test crossed with this recurrent parent and evaluate the progeny in multiple environment with high H square. This H square means the high environmental factors, high degree of environmental effects. That means whether we are trying to select a plant of drought tolerance or salt tolerance, these are the environmental parameter, and we must select these plants with this high concentration of environmental stress. And after that, we have to identify the markers those are associated with the trait of interest. This step can be achieved with the molecular marker selection. I have previously told you that these molecular markers can be identified with the simply gel electrophoresis technique. Then we have to create an index weighting significant markers by their effect using multiple linear regression. We have to I previously discussed these regression can be generated by simply plotting their values on a graph against the molecular marker, and the recombined best progeny or the best individual are picked up from the cycle 0, and these can be easily picked up by the calculation of lot score. The selection of greenhouse or off-season nursery, and these plants those are selected, those individuals are then screened at greenhouse or off-season nursery. That means when the plant season is not, this is not the plant's normal season, we can easily grow a plant in a greenhouse, and this can be cycled up to 3 cycle in low h square environment. That means the environmental stress are low here, but there are some limitations. We generally, in case of Mars, we generally underestimate the number of QTL. That means the we cannot select these traits, those are not counted, and this is the main limitations. Whether we are not counted here, we have to rely on the qualitative traits, and that is much more difficult than selection based on the quantitative traits. And the environmental effects are overestimated here. So the environmental effects may not be um, same according these environmental effects may be changed with the time, and so the estimation is not very up to the mark. Now comes to the genomic selection. Genomic selection is the type of selection which was proposed in 2011 2001. It is proposed to skip the significance test of markers and to use a large set of random markers directly. In case of genomic selection, we use the whole chromosome sequencing and the whole chromosome mapping for identification of markers. And these markers are random, and those are selected directly from the chromosome. The marker effects can be predicted through best linear unbiased prediction. If we choose the markers from the whole genome, then this must be the unbiased condition, and the picking up of the marker is very easy. The genomic selection is a form of marker assisted selection in which genetic markers covering the whole genome are used so that all quantitative traits loci or QTL are in linkage disequilibrium with at least one marker. That means there is a high chance of picking up one gene that is linked with one marker, and there the as we are doing the whole genome scanning, then the chance is much higher. The SNP is the key marker for genome selection. Now, what steps are involved in the genomic selection? It can be achieved by two process. One is the training population. In training population, both the plants are phenotyped and genotyped. The initial parents, those are involved in this training model, are uh, are test crossed, and the test crosses lines are generated. And from the test cross line, the offsprings are generated after nth number of regeneration, and this nth population of offsprings must be transferred into the breeding population. And here the genotype, here these plants are genotype, but no phenotype are considered for this process. This is the breeding population, and after getting the breeding population, the new variety is produced from this breeding population, so that we are now we have now the new variety with the homozygous cell line. So students, this is the genomic selection. Now what are the features of genomic selection? It considers the markers affected effects across the whole genome. The use of high density marker is one of the features of genomic selection. It is based on two distinct and related groups, the training and breeding population. The phenotyping is a key information in GS to build up accuracy of statistical model. This is for the training population. And GS may revolutionize plants as tree breeding practices. These can be used for the tree breeding uh, practices in nursery development. So students, this is the process of genomic selection and marks. I think this will be helpful for your syllabus. So please go through this. Thank you.